Okay, I haven't forgotten about talking about exam 3 a little bit. I'm just not quite able to do that. So hopefully Tuesday, I'll show you a few questions that were more commonly missed. I think there was only 1 or 2 questions where less than half of you missed. So there wasn't really any 1 or 2 that was, you know, oh, everybody missed this and I need to explain this a little bit better, but we still will do that. I haven't forgotten. I wanted to start out, let's see, I had one other thing that, so I had a question about what's going to happen after Thanksgiving break. Right now, I am planning on just recording lectures and posting them, still having lots of Zoom office hours, so I'm available for questions. That's sort of what I had planned in my mind to do, but anyway, if you have any suggestions or things that you think would be helpful, I'm open to that as well. I guess mostly the university is going to be, and the university is technically open, but again, we're encouraged to be all online after Thanksgiving, which really is a pretty good idea, the way things are going. Okay, so I wanted to give you a couple questions from chapter 15, just to kind of warm up your brain a little bit today. Some kind of hopefully straightforward definition type questions. So the structures that allow a phagocytic leukocyte to bind to a PAMP. Remember, PAMP stands for pathogen associated molecular pattern. Okay, what structure is that? That's the TLR, the toll-like receptors. All right, so kind of a, again, straightforward question there. I've had a few of you ask me to go through that again, how that works. Be glad to do that. Again, if you come after class or stop by office hours. The second question deals with the terminology, different kind of antigens. We talked about what makes a good antigen, what typically makes a poor antigen. Again, a little terminology to do with those. So an antigen that is phagocytized by a dendritic cell, what would we call that antigen? By definition, that's something that's outside of the cell and then gets phagocytized. So that's going to be an exogenous antigen. Remember, something that's inside the cell would be an endogenous. So something like a virus, it starts out as an exogenous antigen. Once it's inside of the cell, then we term it an endogenous antigen. And that term is going to be important as we get through today, the different kinds of responses that we have to both exogenous and endogenous antigens. It could be this. We're going to talk more about what that means today, but by definition here, so this is going to be the best answer here. All right, so we ended up last time we had talked about what antibody actually do, the neutralization functions, the complement activation, agglutination, and then we're going through the structure of the different isotypes of antibody. So IgM, first thing I think about IgM, that's the first antibody that's produced when some kind of pathogen is encountered. So we wrote that in. Really a fairly small amount of the total antibody in the blood. Now, if you were to test that, you know, right at kind of the beginning of some infection, like right when antibodies were being made, maybe that would be a little bit higher IgM, but typically speaking, again, that would be fairly small amount in the blood. And if that infection, as the course of the infection wore on, typically the B cells will adjust a little bit and switch to the type of antibody that would be most likely to do good against that pathogen and to clear it. So we call that process class switching. So again, the B cells or plasma cells are pretty short-lived. So whether it's the plasma cells that change the type of antibody or the B cells that become plasma cells, I would have to look that up again. But they change the class of the antibody again, to make an antibody that's even going to be better suited to fight that infection. So typically, the IgM is going to switch to an IgG. Okay, not always, but that's probably the most common thing that it would switch to. So basically, the stem region is changed. The gamma region of the gene is transcribed and is translated to be that IgG. Okay, so this is the most common antibody in circulation. So in the bloodstream, it's a lot of it is going to be IgG, and we think that that's usually the longest acting. So the IgG tends to persist at least a couple of months. Okay? And that's important because it is the only antibody that can cross the placenta. So the mother shares some of her antibody with her unborn child, and then they persist into the first couple months of that baby's life. So again, that's important because young babies don't have a well-developed immune system, so at least they have some antibodies from the mother to protect them in those early days. Now, that can, in certain situations, cause a problem in terms of Rh disease. So that Rh antigen, rhesus antigen, is one of those alloantigens. So it's a different among us humans, some of us have the Rh antigen, some of us don't. So if you're blood type A positive, for example, you have positive for that Rh antigen. If you're A negative, then you don't have it. Okay, so if the mother has the antigen, then it's not a problem for the baby at all, because she's not going to make antibody to the antigen because she has it already. So it would be an auto antigen, and as long as her immune system's functioning properly, that wouldn't be a problem. The problem is when the mother does not have the antigen and the baby does. So typically it's not a problem for first pregnancies. There might be a little bit of mixing of maternal and fetal blood, but maybe not till birth. So if there is some mixing though, then the mother would start to produce antigen. So again, if the mother doesn't have it, the antigen, now she's going to start producing antibodies to that antigen after that first pregnancy. So if she has another baby that's Rh positive, she is going to start making antibodies to that antigen, and that causes the early destruction of red blood cells. So it might just be some mild anemia for the baby, but it could lead to what's called a hemolytic newborn disease that can cause the death of a baby. So in the late 60s, we came out with a Rogam shot, that would mothers take it at a certain point in their pregnancy, and then again at birth, just to neutralize her Rh antibodies that she may have. Okay. All right, so I included on each of these what kind of functions that particular isotype of antibody is good at. For the IgM, it's particularly good at agglutination because it has 10 different binding sites. So that's kind of easy to remember. Probably each of the isotypes do a little bit of all of the different functions of antibody, but sometimes they're especially good at one function or another. So IgG tends to be pretty good at like all those different functions. All right, sometimes IgM, the B cells may switch to making IgA antibody. Again, it depends on the pathogen, depends on what they're fighting. This is the most common antibody in our mucosal membranes. Okay, so very small amounts, again, of the total antibody in the bloodstream, only 12%. But again, on those mucosal membranes, because this secretory component it's a protein that's added to the antibody as it is secreted onto the mucosal membranes. That secretory component tends to kind of neutralize proteolytic enzymes that pathogens may have to try to protect themselves from antibodies. So that secretory component allows them to be there. In the J chain, we saw that IgMs had the J chain already that helps to bind the monomers into a dimer. So on the mucosal membranes, we see IgM as a dimer. If it's in the bloodstream, it exists as a monomer. Okay, so it's on those mucosal membranes. We also find a lot of IgA in bodily fluids. So tears, saliva, and breast milk. Okay, so one more way that the mother can help protect her newborn baby is by passively giving the newborn child some antibodies, some of her own antibodies. Okay, so sometimes students get that mixed up. IgG crosses the placenta, IgA is in the breast milk. So you think baby and you kind of get them mixed up sometimes. Okay, major roles here, neutralization and agglutination. It appears that IgA do not activate complement very well. And that's actually a good thing because we don't want our mucosal membranes. One of the things that complement does, remember, is increase inflammation. We don't want our mucosal membranes to be in kind of a constant state of inflammation. So apparently IgA is not good at that, and that's a good thing. All right, and I don't have a lot to say about IgE isotype and IgD. They're both extremely uncommon in the bloodstream. We don't make as much of them. We think IgE, I think eosinophils, IgE antibody has some of the same function as those eosinophil granulocytes, white blood cells that we talked about. Their main function is to act on parasitic worms. Of course, we're very hygienic in our society right now. We don't come in contact with them a lot. We also associate a higher titer of IgE antibody when a person is having some kind of allergic reaction. Okay, and IgD, we don't know a lot about. We do know that IgD doesn't circulate in the blood. It typically stays bound to the B cell. So it's kind of an antibody that, again, doesn't circulate, stays bound to that B cell. And we don't really understand very much about it. Okay, so in animals, some animals have different isotypes. Sharks have IgW, for example. So there is some different kind of isotypes out there that humans don't have. Okay, so as we move forward, one problem that it stumped researchers for a long time, trying to understand how we get such a huge repertoire of B cell receptors, and of course the antibodies that the B cells make match the B cell receptors. So we talked about how we have B cell receptors that would match up with almost anything that we would ever come in contact with. So stingray venom, for example, you probably have B cell receptors that match stingray venom. Okay, so probably only a couple of them, a couple of cells, but then they can be called into action if they're needed and proliferate and make many more of them. Okay, so we don't fashion antibody, or the B cell receptors aren't made to match a pathogen. We already have them, and then they just proliferate. So the question is, how do we have so many different B cell receptors? Because if we had, each B cell receptor corresponded to one gene, we don't have that much DNA in our body. So how do we generate such a variety of B cell receptors, and in turn, how do we make so many different kinds of antibodies? Well, there was a researcher in the 70s, a Japanese researcher, Tanawanga, I think his name was. He finally cracked this problem. And it has to do with some recombinations that take place in the heavy chain locus. So locus means area. This is the area of the gene. the DNA, where we know that the instructions are for B cell receptors. So, right, we have 2 chromosomes. So each of those chromosomes has an area on the gene, which we call the heavy chain locus. So remember, the B cell receptor has both heavy chains and light chains. And that variable region is right at the tip of those. Okay, so we think in that gene, there's some recombinations that allow us to fashion these unique antigen binding areas, both the heavy chain and the light chain. So the way this works, there's this protein, an enzyme called RAG enzyme that goes down the length of that DNA and randomly picks different segments. So there's a V region where there's 65 different areas. There's a D region with 27 different areas, and a J. So this VDJ recombination, the RAG enzymes goes down, picks one V region at random, one D and one J, transcribes that into some messenger RNA. Of course, the messenger RNA is translated at the ribosomes, and we get this protein receptor. So the analogy they use in your book, which I think is a pretty good one, imagine that you go to your closet in the morning, you have 65 different shirts, you pick one, you have 27 different pants, you pick one, you have 6 pairs of shoes, and you pick one. You get dressed, and your roommate may look at you and say, you are not going out of the house that way. You have pink plaid with purple stripes, not going to work. So sometimes, the point is, sometimes what segments are randomly chosen don't correspond into a workable protein. There might be a stop codon in there for some reason, and that protein doesn't get translated. So this isn't a perfect system, but again, with these recombinations, we get, all these different combinations. And I wrote the math down in here. You don't need to know how I came up with all those numbers, but it is explained in the book there. Basically, with the heavy chain, and again, we've got 2 heavy chains, and then added with the light chains, that gives us almost 10 million different B cell receptors, and we think there's other recombinations or mutations that might take place. So, this is supposed to be 10 to the 23 here. So we've got many different, lots of variability in that B cell receptor, and the similar process takes place for the T cell receptors as well. Okay, and I included this animation. It's just a couple-minute animation, the kind of animation that walks through this. If you want to hear that explanation again, that's a really excellent animation. Okay. All right, so moving on to T cells. I talked about how we need to understand a little framework about T cells before we're going to talk about how B cells actually get activated to make antibody. All right, and we're also going to be talking about how T cells become activated. Okay, so there actually are different kinds of B cells that we're just not going to get into, but the main kinds of T cells, cytotoxic T cells, always abbreviate that TC. Their main function is to kill other cells that are infected with some intracellular pathogen. Okay, so again, inside the cell, a virus is typically going to be intracellular. Okay, and we know that a T cell is a cytotoxic T cell because it has a glycoprotein on its surface called CD8. Okay, so they have CD8 positive. They have these glycoproteins. CD stands for cluster of differentiation, in case you're interested. Okay, I'm not sure where that name came from. And then T helper cells, their main function is they're kind of the general in charge of the immune system. So they're just coordinating many different immune responses. They have the CD4 glycoprotein on their surface. So the T helper cells are a little bit more famous because they're the ones that are damaged by HIV. HIV actually infects a lot of different cells like macrophages, but it doesn't harm them like it does T helper cells. Okay, so if a person is deficient in T helper cells, it's a huge problem, especially in terms of their adaptive immunity. It just doesn't work very well. And there also is a subsets of T helper cells that we're going to talk about later, but it's T helper 1s that are responsible for stimulating the cytotoxic T cells, and it's the T helper 2s that are going to be stimulating our B cells or activating our B cells. So we'll get to that later. There also, I'll just mention something called T regulatory cells. We're not going to really bring them into our discussion anymore. I just mentioned that there is such a thing. Don't really worry about what receptors they have there, but we think that they have some role. We used to call these T suppressor cells. For some reason, they changed the name, but they have some role in dampening the immune response if it gets a little bit out of hand, apparently. Okay, so we talked about how T cells mature in the thymus. Okay, they're going to mature into these different kinds of T helper cells and get their T cell receptors. All right, so if we look at the T cell receptor, that's what's pictured here. And I also included, remember, our B cell receptor just for comparison purposes. You see some similarities. They both have this transmembrane portion here. We've got constant regions, and variable region is where the antigen binds. Okay, but you'll notice here that for B cell receptors, they bind directly with epitopes on antigens. Okay, so that's one big difference that we see. T cell receptors cannot bind directly to epitopes. Okay, they actually can only recognize antigen when it's presented by MHC proteins. Okay, so B cells can recognize antigen on their own. T cells have to have it presented to them in a certain way, and our body's mechanism for that is with these MHC glycoproteins. Okay, so we need to talk about what these MHC glycoproteins are. This first slide, like I said, this is really wordy, but I'm going to talk through a couple of diagrams, and I tried to kind of put the information on this slide so that I'm not writing a whole ton of stuff on the other side. What they are, they're in the surface of most of the cells of our body. And there's two different kinds. There's MHC class 1 and MHC class 2. That stands for major histocompatibility complex, or MHC. Okay, so let me come back to this slide, but I always like to have a visual whenever possible. And this is your book's figure 16.4. Okay, so this one on the left is MHC class 1, and on the right here is MHC class 2. The difference here is MHC class 1 seems to only have, one membrane spanning portion, and MHC class 2 has 2. Okay, but they have different function. The MHC class 1, this is going to present antigen that's normally within the cell. Okay, so MHC class 1 is on every nucleated cell in our bodies. Okay, so Can you think of a kind of cell that doesn't have a nucleus? Okay, red blood cells are the only ones that I can think of. Okay, they of course have a red blood cell and then they lose it at some stage of their development because they don't need it anymore. Okay, so pretty much every cell in our body except red blood cells has these MHC glycoproteins. And their job is to be sampling bits of that cell and advertising on the surface what kind of proteins are present inside of that cell. Okay, so let's see. These MHC class 1, they're going to interact with cytotoxic T cells. It's a little sloppy the way that I wrote that there. Okay, it's the cytotoxic T cells, remember, that are responsible for dealing with intracellular pathogens. So the MHC class 1, that's our mechanism for advertising on a cell if it is infected with a pathogen. Okay, so the way I think of that, MHC class 1, raise your hand, you're volunteering what's inside. You know, I'm going to volunteer the answer. I'm going to volunteer what I know. This cell is volunteering what's going on inside of it. Okay, that we have a mechanism where proteins are kind of routinely chopped up and presented by MHC class 1 all the time. And if that cell is not infected with anything, then those antigens get ignored. But if there is some kind of viral infection, that will be advertised and the cytotoxic T cells can come and recognize that. Okay, MHC class 2 is only present in our antigen presenting cells. So that's going to be dendritic cells, macrophages, and actually B cells can present antigen in a slightly different way than what macrophages and dendritic cells do. Okay, so your book's figure in the newest edition is slightly different than this. I like it, but I didn't have access to it. In the newest figure, it is presented in a way that helps you to understand that these cells, dendritic cells and macrophages, not only do they have MHC class 2, but they're nucleated cells, they also have MHC class 1. Okay, so some cells have both. The MHC class 2 are going to interact with T helper cells. Okay, and then it's so they're going to be presenting antigen that the dendritic cells and macrophages have phagocytized, and they're going to present some of that antigen. So basically, you know, they are helping to get rid of that pathogen themselves, but if their work is not enough, they need to advertise what they're dealing with. And then when they come in contact with T helper cells in, say, a lymph node, then the T helper cells get activated and will act on B cells to start making antigen. Okay, let's see. Okay, so class 2, so if class 1 is volunteering what you know, class 2, you could think about like 2 hands out. Here, you take it. This is what I'm dealing with. That's kind of just a little mnemonic that helps remember which is which. Okay, so let's see, what did I forget to say here? This variation in MHC and the antigen they present play a role in transplant rejection. So basically these MHC genes, that's a polymorphic gene. Is that the right word I'm looking for? I'm not sure if I used the right word there. Basically, we all have different forms of that gene. So in the general population, your MHC proteins match maybe like 1 in 500,000 of the general population. People you're related to probably going to be a closer match. Polymorphism, I think that's the word that I was looking for if I said something different. So we all have different, so basically this accounts, one of the things that accounts for how if you and I were both exposed to the same pathogen and we're both healthy, one of us gets sick and one of us doesn't. Well, we have different ways of presenting antigen to our B and T cells. Your MHC might present, a slightly different amino acid sequence, and our B and T cells maybe will recognize it a little bit differently than the kind of antigen that I present. So again, there are sort of just genetic differences in our immunity that it can account for who gets sick and who doesn't, or how badly somebody gets sick. And they try to match this up as well as they can in terms of looking for organ transplants. Okay, because obviously you don't want to, just to help with rejection there. Okay, and like I said, if that MHC class 1 if that cell is not infected with anything, then those antigens that are presented just get ignored. And, they last for a number of hours or maybe days for a certain time. And then they basically will degrade and new ones get posted, you could say. Okay. So if you think about, well, how do those antigens get there? Again, this is kind of the basic idea. It's more complicated than this, but our MHC class 1, our MHC class 1, again, this is the processing of endogenous antigens. Okay, so any protein that's there in the cell actually gets broken down by an enzyme complex called proteasomes. Okay, so they don't mention that there, but we have a mechanism to do that, to just sample all the proteins in our cell. And the MHC class 1 are made in our rough ER, and then they're expressed on that membrane of the endoplasmic reticulum. Those proteins that are chopped up, they enter through the ER through pores, and then maybe those short amino acid sequences, we're talking like 10 to 20 amino acids, will match up with certain MHC class 1. Those get kind of pinch off from the ER into the Golgi, into this membrane bound vesicle, and then are presented on the surface of the cell. Again, either to be ignored or to call in some kind of immune response, T cell receptors. Okay, cytotoxic T cell. Okay, the MHC class 2 doesn't involve the rough ER at all, because again, this is exogenous antigen. So we have something like a macrophage here, maybe, that's engulfing that exogenous pathogen. It gets chewed up in the phagolysosome. MHC class 2 proteins get expressed on a membrane vesicle that then will fuse with the phagolysosome. Bits of that chewed up, whatever, bacteria cell or whatever, will match with certain MHC proteins. And then the newest edition of your figure posts just a picture. Here's the nucleus of that macrophage, now posting, advertising some of that antigen on its surface here, maybe several different ones. Okay, so again, now this macrophage is going to travel in the lymph to lymph nodes and start making connections with helper T cells. Okay, to call in this is going to be calling an adaptive reaction. Okay, and there is a video tutor here of this as part of your homework. So again, you can visualize that even better. Okay. All right, so it's time to put everything together and to try to understand how T cells get activated. So when that cytotoxic T cell recognizes that antigen expressed by, advertised by MHC class 1, that's actually not enough for that cytotoxic T cell to decide to kill that virally infected cell. It needs some other stimulation. We've got lots of checks and balances, remember, in our adaptive immune system, and we want to make sure that that is a correct response. We don't want our cytotoxic T cells to go killing indiscriminately. Okay, so I've got two versions of how this happens. This first one I found online, and it's just a little bit more basic. And your book adds in a couple more details that I want to call your attention to. So let's start down here. You have this virally infected cell. Okay, so here's the viruses in red here. And by MHC class 1 protein, that's going to present bits of that antigen. And that MHC class 1 is recognized by our cytotoxic T cells. It's got this CD8 right here, cluster of differentiation that kind of stabilizes that immunological synapse. But again, just recognizing the T cell receptor, recognizing that antigen is not enough. It needs some further stimulation from a helper T cell. Okay, so how does the helper T cell get stimulated? Well, it's going to encounter some of that virus that's been engulfed by, say, a macrophage or a dendritic cell and presented by MHC class 2 protein to a helper T cell. Once that immunological synapse is made, then the helper T cell is activated. It starts kicking out cytokines. Don't worry about what kind. And that's going to act on the cytotoxic T cell, sorry, which will then proliferate, make many more of itself, as well as some memory cells. Okay, and now those activated cytotoxic T cells can act and destroy the infected cells. Okay, so once a cell is infected with a virus, it has to be sacrificed. There's no way to save the cell once it's infected with a virus. Okay, so like I said, your book's graphic is a little bit more complicated. I just want to point out a couple of things from it. Instead of showing kind of two different cells, this could actually all happen within some antigen cross-presenting cell, like say a dendritic cell. And it shows this immunological synapse here a little bit better. So you've got an inactive cytotoxic T cell that recognizes this dendritic cell is infected with some, say, a virus. And if we look over here now, it's presenting that epitope from that virus by MHC class 1. Cytotoxic T cell recognizes that. The CD8 recognizes the MHC class 1. But again, just this immunological synapse, that's not enough to activate the cytotoxic T cell. It needs signals from T helper cells as well. So over here, we've got a T helper cell that recognizes the same epitope from a virus that was outside of the cell and got engulfed by a phagocytic cell, presented. Now the T helper cell with correct stimulation is going to differentiate into a T helper 1. All right, so if you think T helper 1 is going to help with the MHC class 1, which is cytotoxic T cells. Again, the T helper 1 cell releases the cytokines that then will activate the cytotoxic T cell into clonal expansion. That just means it makes many more of itself, as well as some memory cells. These memory T cells, the next time it encounters some epitope like this, it doesn't need additional stimulation from T helper cells. It's ready to go. Okay, so you get a few memory cells made. The cytotoxic T cells basically stimulate themselves by releasing certain cytokines to make many more of them. I was looking at this picture really closely last night, and I thought, why does it look like these cytotoxic T cells are smoking a cigarette? That's what those look like to me. Those are receptors for cytokines. Okay, so it's got to have a receptor for this IL-2, and then when it releases the IL-2, again, it's self-stimulating. Okay, so then you get a whole lot of activated cytotoxic T cells. Okay. What do the cytotoxic T cells do? Okay. So once a cytotoxic T cell is activated, okay, so this is activated. Once it comes in contact with a virally infected cell, okay, here's again the MHC class 1 that's presenting the epitope. Here's the T cell receptor and the CD8. Once that synapse is now made with an activated cytotoxic T cell, okay, then the activated cytotoxic T cell is going to start pumping out a couple of cytokines that are going to act to kill this virally infected cell. Okay, so you can, this figure is from your book there. If you, it's a good exercise just to kind of label all those parts yourself. Okay, this is one of the ways that a cytotoxic T cell will kill a virally infected cell. There's a couple different ways. We think this way is a little bit more common. There is like a paragraph about the other way in your book. I'm not going to go into that other way. I want to talk through just briefly the perforin-granzyme cytotoxic pathway. So again, this is a cytotoxic T cell that's activated. It starts releasing two cytokines. One is perforin, and what perforin does is it makes holes in the membrane. So we'll say pokes holes, or you could say perforates in the infected cell's membrane. Okay, remember, this is one of our cells that's infected. It's no cell wall or anything like that. So pokes holes in the membrane, which in itself is bad, right? Having holes in the cell membrane is going to eventually be bad for a cell. But it releases another cytokine called granzyme that then it enters into the host cell, and it induces a programmed cell death. We call that apoptosis. Again, this is like a cell suicide, programmed cell death. All right, so I'm sure there's a whole mechanism that we could get into how that happens exactly, but just recognize perforin pokes the holes, and then granzyme induces the cell death. So again, have to sacrifice that host cell when it gets infected to try to keep that virus from lysing the cell and releasing many more virions. All right, questions? Okay. All right, so let's move on to B cells. Looks like messed up a little bit of my indenting here. That's okay. So there's actually two different ways that B cells can be activated. In your book does not, only mentions really briefly the T cell independent antigens. I like to talk about that because of a particular point that I want to make about that, and that I'll get to that in just a minute. But basically, certain antigens are able to activate B cells all on their own. So we call those T cell independent antigens. They don't need T helper cells in order to activate B cells. So the terminology throws people sometimes, like if you remember, when we were talking about how transcription ends, Sometimes it involves Rho protein, sometimes not. So the Rho independent or Rho dependent pathways, sometimes just thinking through what those words mean exactly. Okay, so again, in this activation method, T cells aren't necessary. Okay, they're particular antigens that will activate B cells all on their own. What kind of antigens are able to do this? Typically, they're pretty large molecules with lots of repeats. Like there are some polysaccharides that are able to do this. You remember last time we said that usually polysaccharides aren't very antigenic because it's just the same thing over and over and over again. There are exceptions. Okay, that's true in a general sense, but there are exceptions to every rule in biology, it seems like. Okay. The nice thing about these kind of antigens, I guess, in terms of our response, is that we're able to quickly mount a response because you don't have to have all this mixing in the lymph nodes and finding, you know, all these different interactions have to happen. It's just the antigen itself activates the B cells. So we get a pretty fast response. The problem is, in terms of fighting these pathogens, is that we have a weaker response when it's a T cell independent antigen. Why is it weaker? Well, it doesn't produce any memory cells. So even though it's a little quicker response than our other method of activation when T helper cells are necessary, we don't get memory. So that's sometimes an issue, and there's no maturation of antibody. I have that defined on a slide that we'll get to pretty soon. Basically, that means in a typical course of infection, we get some point mutations on the variable region of the antibody, that the antibodies over time tend to fit the antigen a little bit better. We don't see that with a T cell independent antigen response, and we don't get any class switching. So typically IgM antibodies are produced first in a typical infection, and with this T independent, typically that's all you get is IgM. So there's no kind of fitting a best response to that pathogen. Okay. And another problem, I should indent this because this is certainly maybe the biggest problem, is that young children mount a very poor response to these kinds of antigens. Okay, so because of the just naivete, I should say, of the immune system, it's just not well developed. Again, young children don't have a very good response. So one pathogen that is considered a T-independent antigen because of the capsule that it produces is this Haemophilus influenzae. You may think this causes the flu. It's actually misnamed because back during the pandemic of 1918 and 1919, we thought this bacteria caused influenza. That was our best guess. We don't really know what caused that pandemic. So that had to be kind of a scary thing. But we thought it was this bacteria. Actually, we know now that this bacteria causes meningitis. So this is bacterial meningitis is a fairly scary infection. There are other organisms that more commonly cause meningitis, what we call a meningococcal meningitis. But we never see this infection anymore because we have a good vaccine to Haemophilus influenzae. It's the Haemophilus influenzae Hib vaccine. You might have seen that. Okay, so it's Haemophilus influenza type B. All right, so this is the main reason I wanted to talk about T-independent antigens, because it's vitally important that babies get their Hib vaccine. And again, parents may think, oh, I never heard of that. Nobody ever gets that. That's because adults don't get it because we have a good immune response to it. And we almost never get it, but babies are very susceptible to it. So this is, again, typically one of the first vaccines that babies get, and it's important because obviously you don't want your baby to get meningitis. Okay. So here's the graphic that, again, used to be included in your book. You won't find that in the newest edition. But typically T-independent antigens are some polysaccharide with repeating subunits. Not every polysaccharide, just there's a few of them that act as T-independent antigens. And again, the recognition of B cell receptors to that antigen causes the B cell to change into plasma cells, which then start pumping out the antibodies. And again, typically these are IgM, even though they're not drawn that way there. Okay, and notice what we're missing here, no memory cells. Okay, so that's again, a drawback. Okay, how are B cells more often stimulated or activated? That's by what we call T cell dependent antigens. So the B cells rely on T helper cells for a second signal before the B cells will start differentiating into plasma cells to make antibodies. Okay, so again, as part of, let's see, what did okay, I think I posted this on this page here, but that is another great animation of B cell T-dependent activation. So again, I would watch that at some point, but I'll walk you through this here. So here we have some antigen presenting cell. So again, dendritic cell, macrophage. Here's a blow up of that immunological synapse where that antigen presenting cell has phagocytized some antigen and is presenting a certain epitope with its MHC class 2. Remember, it's the T helper cells that recognize MHC class 2. They've got that CD4 glycoprotein. Don't worry about these other CDs. We're not going to go into that, but there's some further stabilization of that immunological synapse. Okay, but so now the T helper cell is activated. At the same time, you've got some B cells that are ingesting antigen and presenting it by MHC class 2 as well. Okay, so B cells don't really phagocytize, but they are able to, again, kind of encircle antigen in an endosome and present it by MHC class 2. Okay, so then these B cells, here's a blow up of this synapse. What I forgot to say is these T helper cells up here, here's the T helper cell. Once that has made this connection, it will differentiate into a T helper 2 cell. Okay, and that activated T helper 2 cell, if it comes in contact with one of these B cells over here and recognizes, again, the same antigen, now cytokines are produced that are going to activate that B cell into plasma cells and antibody secreting cells. Okay, also get some memory B cells that are going to circulate for much longer. And the next time these memory B cells recognize that same antigen, they don't need that T helper 2 cell stimulation. They can start making antibodies all on their own. Okay, questions? Okay. So definitely check out that animation. So basically, what happens in terms of thinking about the first time we encounter an antigen, how does our adaptive immunity get called up? And comparing that to the second time we encounter some antigen. Okay, so this is kind of basis of vaccines we're getting into here. So the primary response, again, that's the first time your body comes in contact with some antigen. So let's say, for example, you get a tetanus vaccination. Okay, our tetanus vaccine is actually, it's called a toxoid. It's the tetanus toxin that's been inactivated. Okay, so say a patient is given the tetanus immunization. There's a bit of a lag period because that protein has to have time to get processed and presented, travel through the lymph, find the correct T cells and B cells, all those interactions that get made typically in a lymph node. So eventually our bodies will start pumping out some antibody. Usually takes about a week for that to happen, maybe, in full amount here. It's kind of hard to read the time scale there, but maybe 6-7 days. Okay, so IgM is the first antibody. Over time, the response gets a little bit better. We get some class switching, isotype switching. Typically might get some IgG, a little bit higher amount, until that response is basically the danger is past, basically. Okay, so once we're immunized with tetanus, and say hopefully we have that done about every 10 years because it wears off after about 10 years, say 5 years later that patient encounters the real tetanus bacilli. Maybe tetanus is more likely to be contracted if you have some kind of puncture wound, although you can get it from any kind of scratch, but it's an anaerobic organism, so kind of a puncture wound is more likely. So if you were like my cousins who were playing catch with a pitchfork when they were about 8 or 9, pitchfork went right through one of my cousin's foot. They took him to the hospital, of course, and got the wound taken care of. And they said, well, you need a tetanus shot. And he said, no, it was a really clean pitchfork. I don't need a tetanus shot. So anyway, they're probably going to give you a tetanus shot, even if you've had one recently, if you have that kind of a wound, just to make sure you have a good response to that. But say at some point later you are exposed to the actual toxin, because you've been immunized, you're going to have a much quicker antibody response because you have memory B cells for sure, maybe T cells anyway. Also, you're going to have a much quicker response, and that class switching happens right away. You get even more IgG much faster. Okay, so more antibody, the right kind, much quicker. The second time you encounter an antigen, third time it might be an even better response. Okay, so again, this is the basis of why we immunize. So we have such a fast response that we don't even know that we've been infected with some kind of pathogen. Okay, so again, this would be a T-dependent response. Okay, so here is from another source, basically the same kind of graphic. Primary response, if say it looked like this on the left, second time somebody encountered that antigen in a T-dependent antigen, okay, we'd get more IgG, probably even a little faster. What about the primary response and the secondary response if you're dealing with a T-independent antigen? Okay, what do you think our secondary response would look like if this was a T-independent antigen? Same, maybe? Oh, well, so you mean the same as this or the same as the primary? Yep, so good. So I didn't post this, but right, for a T-independent antigen, remember we said there was no memory, there was no class switching, so it looked the same the second time. It is a little faster response, so we have that going for us, but very different secondary response there. Good. All right, so, oh, one more thing that we need to talk about as we finish up our adaptive immunity. Okay, we did a lot today. But we also need to think about, okay, so if we have this huge repertoire of B cell receptors and T cell receptors, what is the mechanism so that we don't have B cells and T cells that target our own self antigens? There has to be some kind of mechanism. If they're made randomly, certainly some of them are going to get made that recognizes our kidney cells or our hemoglobin or our muscle cells. We have to get rid of those so we don't have autoimmune reactions. All right, so for T cells, remember T cells are made in the red bone marrow, but then they travel to the thymus in bloodstream in order to undergo further maturation. So I talked about, well, what does that mean? That means they mature into different kinds of T cells and their T cell receptors get made. We also are going to get rid of the T cells that are going to recognize, again, our self antigens. So the way that happens, so here he's got a repertoire of T cells in the thymus. So in the thymus, there's two tests that have to be passed. Basically, we need T cell receptors that recognize our MHC. Okay, so remember I said, you know, MHCs, there are a lot of different varieties of that. We want our T cells to recognize the MHC. There has to be some recognition of that. Okay, so they go through a process in thymus cells. It's not really well understood, but thymic cells have a way to present all different kinds of proteins from throughout our body. Okay, so again, the kidney cells, the muscle cells, they present snippets of that to see if the T cell receptors recognize it. Okay, so number one, the T cells need to recognize the MHC. If they don't, then they're allowed to die. Okay, we don't want those. If it does recognize the MHC, so what's not drawn here is there's going to be some kind of, you know, recognition like this to recognize the MHC. If it does recognize the MHC, then it receives a survival signal, some kind of cytokine probably. Then the second test is, does it recognize the MHC complex with the self antigen? And if the answer is yes, then most of them, again, are allowed to die. We don't want T cell receptors that recognize our self antigens. Okay. We want ones that will tolerate ourself, that will ignore ourself. Okay, so if they don't recognize our self antigen, they're allowed to survive. Okay, a few cells over here are allowed to live that function as T regulatory cells. And again, I don't really understand totally how those function, but we do have a few of those. But this repertoire of immature T cells then are most likely to be the ones that recognize some kind of pathogen. It's really important that lymph doesn't come into contact with the thymus because we don't want to present bits of pathogens to these immature cells because then they'll learn to recognize those. And any T cells that recognize, you know, exogenous antigens, we would get rid of those. So if we learn to recognize them, then we wouldn't have any adaptive response really. So it's really important that the thymus is not in contact with the lymph at all. Remarkably, our thymus starts to kind of deteriorate after birth. So most of this T cell maturation is actually taking place even before birth. So we can lose our thymus as adults and it's not a problem. All right, a similar thing takes place for B cells. It's a little bit less complicated because they don't need to be presented antigen with MHC class 1 or 2. The stem cells in the red bone marrow undergo, you know, the maturation process here stays in the red bone marrow. And basically, though, B cell receptors are presented with the self antigens the same way. And if they recognize a self antigen, then they are allowed to die. And if they don't recognize self antigen, they actually travel in the bloodstream to the spleen, where they go through a little bit more maturation process. So there actually are some different subsets of B cells that we haven't talked about. But it's a little bit, again, a little bit more straightforward. There is some second chances here. Basically, if there is a autoantigen recognized, at least with B cell maturation, sometimes they're allowed to go back and change their receptor a little bit and kind of try again. But so everything's a little bit more complicated than what we talk about. We're trying to master the basics here. All right, so hopefully we don't have B cells and T cells that will attack our own body. Okay, when something goes wrong here, then that's one of the reasons that we could have autoimmune disease. All right, so it's kind of a segue into talking about viruses. We have some terminology that we use to describe how we acquire immunity to a disease. And again, I got a little crazy with my pictures there, and when I transferred it into my different programs, some of my words got covered up. Should be right in your notes there. So again, different ways that we acquire immunity to an infection. The terminology here, either we acquire it naturally or artificially. So if you acquire it naturally, the question I ask myself is, could we do this 500 years ago? If the answer is yes, then it's probably natural. If it's, involves some kind of technology or a syringe, then it would be artificial. So I'll give you some examples of that. And then, do we have active immunity or do we have passive immunity? If we have active immunity, that means we're making our own antibodies. Passive immunity, the patient is getting antibodies from another source, okay, preformed antibodies to help them fight off infection. Okay, so look at some examples there. If a patient has naturally acquired active immunity, that means they got that infection, their body fought it off, and now they have memory cells and they won't get it again. So just because you have an infection doesn't mean you'll never get it again. It depends on the pathogen, but there's a lot of things like chickenpox, for example. Usually you get it once, you produce good memory cells, and you're not likely to ever get chickenpox again. Okay, so normal life experience, you're exposed to the pathogen, you have good memory produced from that, and you don't get it again. Natural passive immunity. So passive, that's you're getting protection from someone else. And the only example I could think of there is breastfeeding. All right, so babies are getting those IgA antibodies from breast milk. So passively, the babies aren't making their own antibodies, they're getting it from the mother. And they're only gonna last, a certain amount of time, a month maybe. Okay, but they get that protection right away. The antibodies are functional to, neutralize or whatever. An artificial active immunity, anytime you get an immunization. So, did they do that 500 years ago? No. Okay, so that's artificially inflicted. It's not through like normal life processes. But the patient then is able to make their own antibodies because, again, they have this whole reaction to the vaccine. And then artificial passive immunity, we also call that immune therapy. So sometimes if patients are deficient, maybe they can't make a certain kind of antibody, maybe have a genetic problem there, then patients can get antibodies, again, through an IV. People have to go in maybe every few months to get some antibodies just so they're not getting sick all the time. Okay. All right, other questions? Okay. So again, you can be working on both chapter 15 homework and chapter 16. The 16 is extensive. I think the book says it'll probably take like 80 minutes to do it, so a little more than an hour. Again, put your time in, watch those videos, get out of it whatever you can. Probably a good time to stop for today. I think we're doing good as far as time. So next week we'll start with the vaccines, and we'll also have a chance to get in staphylococcus. Probably it'll be vaccines and staphylococcus. That'll be the end before your next exam. Question?
Do you think it will go into chapter 19 on the exam?
Yes, I anticipate going into chapter 19. It's good to have a little material that's not immunology. It's a little bit more straightforward. So, yeah. Okay, so have a good weekend, and stop by my office hours tomorrow by Zoom.